All right, there we go. The recording started. So last time we left off, we had just finished talking about this first group of protozoans. But I want to step back for just a second and kind of make sure that I emphasize a point that is very important to emphasize. This whole idea of a kingdom protista is really, really sketchy, okay? There really isn't anything. It's still lumped in the kingdom protista, all these critters that we've been talking about or going to talk about. But it's understood that that's a temporary condition. That the kingdom protista is really, as I said the other day, is polyphyletic, right? There's all kinds of different things that we call protozoans, and they're actually more closely related in many cases to things that aren't protozoan-like at all than they are to other protozoans, right? So, like, for example, the red algae that will, and we might get to red algae today, I'm not sure, but a red algae is a protozoan, and a red algae, if you trace back this cladogram, it's actually more closely related to land plants. You see, they share a common ancestor back here than it is to all the protozoans we talked about the other day and the ones we're going to talk about today. And then the choanoflagellates down here near the animals and the nuclearids, those protozoans are actually more closely related to fungi and animals than they are to other protozoans. So that's what we mean by the protozoan kingdom being artificial. It's a kingdom of convenience and being polyphyletic. I'm sure you remember what that term means, right? So that's why the protozoans or the protista group is considered the stem eukaryotic group. These were the first eukaryotic, unicellular, sometimes kind of multicellular a little bit, but all the other groups of organisms, all the more prominent, if you will, only because we see them, the land plants, the fungus, and the animals, arose out of this stem of imagine this protozoan soup, this soup of different kinds of protozoans. There were the excavata, the stromatophiles that we'll talk about today, the amoebozoans, and this group that fits in with the land plants. That, there's this word algae again. See, there's red algae and green algae and golden algae, and some of the algae are prokaryotes. We saw the blue-green algae, and some of these algae are protozoans, and then there are some green algae that are kind of considered plants. So remember, it's what we were talking about early on in the semester. When we talk about the different species, it's not always so clear and sharp, is it? It's not like horsey, a cow. Sometimes it's horse, donkey, mule, dog, wolf, wog. It's that thing where, like, see if I get a little bit of light. I look at myself in the camera there. It looks like I'm sitting in a morgue, but it's getting dark outside. So anyway, if I lose connection, we talked about that when the semester started. Just hang on, and I'll be back. So that's why when we get this idea that these things aren't set in fast categories. These are things that are evolving and changing. Even as we speak, they're evolving and changing. All right, so we went through the excavata last time, the diplomonads, the parabasalids, and then the euglenozoas. I want to jump now into the sarclade. And notice that these groups, the excavata, the sarclade, the archaeoblastida, and the last one at the bottom, really cool, uniconta, is that how you say that word? The uniconta are all groups of protozoans. They're not necessarily taxonomic classifications. Things that have taxonomic classifications like, chordata and mammalia are typically considered official, if you will, even though I just said the kingdom protista is not official. So these are still groups of protozoans. As far as relationships, in some cases, we're not exactly sure about that sort of stuff, okay? So let's jump in now into the sarclade. Let me find where that starts because... Okay, so this is the endosymbiosis. The thing I want to point out again about the endosymbiosis, I was looking through these slides before class, and I think I forgot to mention, and it's thundering and storming outside, folks, so if I may lose connection here. Anyway, what a nucleomorph is evidence of endosymbiosis, because this is an actual picture, an electron microscope picture of a living cell, and a nucleomorph is a nucleus of a cell inside of another cell. See, this is a cell inside of a cell. So that's direct evidence of endosymbiosis, right? The nucleomorph there actually has its own DNA. And in this particular case, the nucleomorph in this picture has DNA that says it's actually a green algae. So like you see in the secondary endosymbiosis up here with the green algae, there's direct evidence that indeed that occurred. All right, Excavata, baselids, euglenozoas, these guys, the sarclade. Now the sarclade, SAR stands for something. It's an acronym. It stands for the Stramenopiles. So I'm looking at the black rectangle on the left. The Stramenopiles, the alveolates, and the rhizarians. We're going to talk about each one of those, and then we'll break them down and talk about each one, that you see in the yellow boxes, the diatoms and the algae. The sarclade is in a very large group, but because of genomic sequencing, we've decided that, or looks like the sarclade is monophyletic. So these dashed lines down here, at least if nothing else, this line ought to be solid right here, letting us know that everybody in the sarclade is, we have, the ancestor and all the descendants. So we think we've got that. How that fits in with the rest of the protozoans, well, that's a whole different story. And what is the relationship between the Stramenopiles and the alveolates? Okay, see, so all these pieces fit in a little bit at a time. The sarclade, like we were looking at secondary endosymbiosis, a lot of the members of the sarclade engaged in this secondary endosymbiosis, where you basically engulf something that engulfed something before. So let's go with the Stramenopiles. We'll look at that as a group, and then we'll break it down into the diatoms. It always helps to me if, especially with unfamiliar words like Stramenopila, right? Not a word that we see or use every day. If we can break it down into the roots. So stramen means straw, and pila means hair. Remember, pili. Between the prokaryotic cells, they could make that tube. When they were conjugating, we call it a sex pili. So a pili is a tube, if you will, or a hair, basically. Stramenopila get their name because if you look at the flagella, they have hairy flagella. Now, of course, it's not really hair. You can see the bigger picture down below. It's kind of like mini flagella on other flagella. But that is characteristic of members of this group. And oftentimes, they'll have a hairy flagella and then another smooth flagella. I think that picture is interesting because if you look on the left where the actual Stramenopile is, the organism, it looks like one of those shower scrubbing things that you use in a shower. Instead of a washcloth, you put your soap on there and you know what I'm talking about? You guys agree? Is that what it is? Oh yeah, or that. That's true. Now, there are some Stramenopiles out there that do not have hairy flagella. Okay, and that's a good illustration of the fact that we've got to remember that when you're talking about organism relationship, if you want to put them into real groups, you got to keep in mind that the way they look today, at the end of that branch of the family bush, if you will, may not be where they came from. So the Stramenopiles today that don't have hairy flagella at one time had hairy flagella. All right, so it's kind of like that everybody could photosynthesize that we looked at the other day, then some folks lost it. It's kind of the same thing with the Stramenopiles. So even amongst these protozoans, there's that difficulty with just because it looks alike or, or in this case doesn't look alike, doesn't mean that it doesn't mean that it's not related. If you're wondering what that is in the background, I am getting some thunder, so you might be hearing that. Oh, these are cool. Oh my God, the diatoms. So the diatoms fit in the Stramenopiles. That's a member of this group, right? Over 100,000 species of diatoms, all of them, well, not all of them, but because many species look alike, but they have all these wide, bizarre little shapes. I mean, this is not computer generated. These are actual pictures. I mean, the coloring has obviously been modified, but the pictures you see in the background and the purple one you see in the right and the little pictures you see in the left, these are all actual diatoms, the neatest organisms. 100,000 species, folks. To put it in perspective, I think there's 30,000 species of fish, somewhere in that vicinity, something less than 10,000 species of mammals. So this is what we call an extremely species diverse group. But photosynthetic unicellular critters with shells You can even see it looks like it's got a shell made of silicon dioxide. All right, silicon dioxide, which is one of the major components of sand. Okay, and we'll talk in just a second why this silicon dioxide. The shells fit together like a shoebox. If you looked at it from the side, well, blue will work. I was going to change the color. The top shell of the diatom fits over top of the smaller bottom shell, or other way around. There's really no top and bottom. And when they reproduce, what they do is they basically, the shells split in half, and then each one just completes the other side. Cool. You can even see in this picture that it kind of looks like this is the silicon dioxide shell. All right, a shell, or sometimes a shell is also called a test. I don't have it on the screen here, but you may see that word, a shell or a test. It means the same thing. Okay, a shell of silicon dioxide implies that there must be some need for strength in these shells, so that these organisms and they're marine organisms, I think there are a few freshwater species, but many of them marine species live in the ocean. So, you know, with this shell that seems to indicate there needs to be some strength, it's one of the sort of the best things about biology and science is the detective work you get to do. So if you look at the diatom and you go, why does it have a silicon dioxide, kind of a glass shell? And relatively speaking, that's a fairly rigid substance, right? Well, that's because a lot of these diatoms, many of them are found in very deep, where the pressures are incredible. That's why it needs such a strong shell. So just to give you an idea, diatoms can withstand pressures up to 33 million feet per square foot. Now let me give you a sort of a sense of perspective. When we're at sea level, and here in Florida we're about at sea level, the column of air that's pushing down on us, and air does weigh something, it doesn't weigh much, but all the way up to the top of the atmosphere amounts to about 16 pounds per square inch. Now, I don't know what the conversion is between square inch and square feet, but you can imagine that a square foot is obviously bigger than a square inch. Okay? In the ocean, every time you drop in depth, every time you go in depth, 10 meters or a little over 30 feet and some change, you add another 16 pounds per square inch of pressure. Okay, we call that 16 pounds per square inch that you feel at sea level. We call that an atmosphere. That's one atmosphere. So it takes, miles of air above you to have one atmosphere of air pushing upon you, and you go down 30 feet, just a little further than 30 feet in the water, and you've got a whole nother 16 pounds. So now you're at 32 pounds per square inch, and then another 30 feet, and you add another 16 pounds. You see the pressure builds up very quickly in the water. You only got to go down 30 feet before you've added a whole nother 16 pounds or atmosphere of pressure pushing on your body. So when you get down to the bottom, and what's the deepest part of the ocean? How deep is the deepest part of the ocean? Does anybody know? Let's see if we can wow you with this. Yeah, it's about 36,000 feet, right? So, and next time you're flying in an airplane, and it's at cruising altitude, and airliners cruise about 36,000 feet, look down, okay, look down to the ground and imagine that's what the bottom, and Dustin, do you know where that 36,000 foot depth is? You know what that's called? What area of the earth? There you go. You're looking down, excellent. You're looking down to the Marianas Trench there, and looking all the way at the bottom. That's what it looks like if you were on a ship floating on the surface of the ocean, looking down to the bottom of the Marianas Trench. That's how far down it is, to the bottom of the ocean. There have been folks who've gone down there. Back in the Ballard went down not too long ago, and back in the 50s, the original folks who went down there. The pressures, 33 million pounds of per square foot. James Cameron, right? Exactly. Oh, Ballard did the Titanic. That's right. If you ever get a chance over in Vero Beach, stop by and take the tour of the Harbor Branch Oceanographic Institute, HBOI. If you put that in to Google, it'll pull it up. It's acronym there. It's an oceanographic research facility, but it also has tours, and they have these really cool videos they show you when they're underwater with the submersibles. But one of the things you'll see, they have a display of these styrofoam cups, like I've got in the picture there. The styrofoam is mostly air, right? So what they do, just as kind of a way to show you the pressure, is they take the styrofoam cups down in some of these submersibles, like in this one, the Deep Sea Crush, or they take them down in some sort of a probe, and the water pressure just presses that styrofoam cup together and takes a regular styrofoam cup and shrinks it down to like the size of a shot glass. It's really kind of impressive. And Harbor Branch has got a collection of those sitting up there at certain depths. You guys are very aware of what I'm talking about, right? Because even if you dive into a pool, to the bottom of the pool, you can feel the pressure on your ears. And if you're a scuba diver, the deeper you go, even snorkeling as well, that's why that thing is in your mask, so you can hold your nose and equalize your air pressure. And that's why you always have to be careful when you're coming up from scuba diving. You never want to rise faster than your bubbles, your exhaust bubbles that are coming out of your regulator, because if you come up too fast and you release that pressure too quickly, it can affect not only your eardrums, as you would imagine, but also can give you what's called the bends, which is nitrogen gas in your bloodstream. So it's not something to mess with. There are actually organisms that live in this kind of environment, not only diatoms, but fish, species of fish. Flatfish, get it? Flatfish. Actually, there are flatfish down there. If you are an aquarist or you have an aquarium, you may have come across this idea or come across this thing called diatomaceous earth. Diatomaceous earth is a filter material. That's why they use it. It's basically ground up diatom shells or diatom tests. And so it's basically powdered glass. It's great for filtering out particulate material and other pollutant kind of stuff in aquariums. So it's very often used for that. A carbon sink. Diatoms can soak up a lot of carbon, especially when there's these big algae blooms that occur. Something that's a sink is some place that something goes. Okay, so like for example, we could call the ocean a heat sink. So that when the sun comes up and starts heating up the earth, the ocean absorbs a lot of that heat. Okay, that's why land stays relatively, our fluctuations of temperature night and day aren't that great because we have so much water. So during the day when the sun comes up and you would think the planet get really hot, well, the ocean is a heat sink and absorbs a lot of that heat. So the land doesn't heat up. And at night, when the land you would think would cool off very quickly because of no sun, then the ocean is actually a heat source and starts releasing that heat. So things are sources and sinks, and diatoms can act as a carbon sink during algal blooms, during times when there's just bunches of them that can actually absorb carbon from the environment. Neat critters. Look up some diatoms online. Look at all the different shapes. They're just really cool. I mean, again, I don't think the purple coloring is natural. That's probably something done with a computer, but just the shape alone. Oh, here we go. Here we go with that term algae again, right? These are the golden algae, and we have, like I said, we have prokaryotic algae. There are plant algae. Now these are protozoan algae. They're called golden algae because of they're photosynthesizers. Some are mixotrophic, right? So they can photosynthesize and do heterotrophic, but all of them can photosynthesize at a minimum, and they don't use chlorophyll to do that. And if you remember back in Bio 1, the pigments for photosynthesis, there was chlorophyll A and there was chlorophyll B, which are both green. There was a xanthophylls, which maybe you talked about. That one's kind of brownish in color. And then there were carotenoids. Does anybody remember what color were carotenoids, and what food do we find lots of carotenoids? Carotenoids, yes. We're still in Instrumentopila. Carrots, right? So what golden algae get their name from the yellowish orangish color of the carotenoids that are in them, okay? Like this Dinobrine, if you look at it, it kind of looks like it has a golden tint to it, doesn't it? critters in this group are all biflagellated, so they have two flagella. All right. Now that becomes important later on when we start looking at different groups of protozoans that break off, because one of the things that begins to differentiate the groups of protozoans and stem protozoans that give rise to other organisms are the number of flagella, whether it's on the front or the back. If you think about it for just a second, and you think about even in ourselves, in us humans, there is a stage that we create that kind of looks like a protozoan. Males especially, obviously, right? The sperm cell with its posterior singular flagellum is indicative of other organisms with singular posterior flagella. You see, like we were talking about with developmental genes, when organisms have similar developmental stages, or when organisms have similarity of these stages early on in their life and developmental stages like I'm talking about, it oftentimes indicates a relationship. It oftentimes indicates a close evolutionary or taxonomic relationship. So, golden algae are biflagellated. So we're going to be talking more as we move on about other organisms that are uniflagellated. As a matter of fact, that word uniconta, conta refers to a flagella. Una means one. All right, so that's significant about that. Most of these guys are unicellular in the golden algae, although there are some colonial versions, which again, what makes the difference between colonial and multicellular and all that, we've talked about a number of times. There's no set line between it. All right. It's very interesting because the evolution of multicellularity was so important to the evolution of organisms on Earth. And like everything else, it didn't happen at a time. It kind of slowly happened. So organisms, even back in the prokaryotes, we had biofilms, right? And biofilms were kind of collections of organisms that were dividing up the labor and kind of working together, right? Many golden algae form cysts. Now, if the environment deteriorates, so if the things start going bad, it forms cysts. Why do you think that is? What do you think the purpose of the cysts are? Yeah, kind of a hibernation, right? Remember, we talked about it in anthrax with the endospores, and I mentioned with this idea of spores, and cysts are kind of like spores. They're resistant structures that can deal with harsh environments. You see, it's sort of like loopholing your way around evolution, or loopholing your way around natural selection, really. Instead of adapting to the environment, what you do when you form a cyst or a spore, and golden algae and anthrax and aren't the only ones, fungi do this, lots of other organisms do this. What you're doing is you're making this resistant structure and hoping that sometime in the future, you're going to make a lot of them because most of them are not going to survive or last that long. Hope that sometime in the future, the environment becomes conducive again to growth. So a cyst is kind of another way, instead of dealing with adapting to the environment, you wait out the environment, okay? Now, there's going to be evolutionary and natural selection trends that lead to the development of successful cysts and spores. I mean, the whole idea is at least some of you come out on the other end of this deteriorating environment, right? It's all about survival. Yeah, Bradley, that's right. So cysts, spores, all that sort of stuff is going to become a really sort. And if you think about what's going on, remember I said this class is an evolution class. And if you think about basically what we're talking about in organisms is how they've adapted and changed through time. So if you're talking about critters that might have been some of the ones that were related to the critters that came out of the water onto land, that went from that relatively cushy environment of water, okay, to the really harsh environment on land, then making these things like spores and cysts and numbers, making lots of them, those are great survival techniques, aren't they? If you know most of them are going to survive, you're going to make a lot of them, and then you're going to try to do things that you can to kind of make these cysts and these spores, and they do. They sometimes cover them with things, resistant structures. There's some spores out there that are so durable, you got to boil them in acid to get the covering off of them, right? So that's going to last a while. All right. One more Stramenophile, and this is another algae. This is what we call the brown algae. All right, so there we go with that algae term again. And a brown algae is a protozoan. Things we call kelp or brown algae. It says here all are multicellular. Again, I want you to take that word with the understanding that multicellularity is not an absolute, that it is a relative term. All right. So right away, when you look at this, you realize something about the term that we've been using all along. All along, we've been calling these things seaweeds, right? But a weed is a plant. These things aren't plants at all. Now, honestly, before class today, when you looked at pictures of these kelp forests, that's what that is, did you think or did you know that those weren't plants? Did you think they were plants when you looked at a kelp forest today? Well, some of you had, if you had a marine science class, you might have known that, but I know when I was in college and first time I took a marine science course, I was surprised. I didn't realize. I thought that they were plants. We called them seaweeds. My goodness, they look like plants. They grow like plants, don't they? Look at the next picture. This is laminaria. There you go. This is another brown algae. Kelp is one. Laminaria is a type of kelp, but this is laminaria. Look at laminaria, the way it's growing. Doesn't that look like a plant? Yeah, it looks like an underwater palm, doesn't it? Laminaria palm, absolutely. Okay, so and you'll notice that it looks like a plant, but it isn't a plant. So we have to be technical about this. So you see, the thing that holds the brown algae onto the surface is called a holdfast, not a root. Roots are terms that technically are referred to only to plants, true plants. So this thing can't have roots. It doesn't have a stem, it has a stipe, and it doesn't have leaves, it has blades. Now, you'll oftentimes see when they're talking about brown algae and kelps, you'll see the term roots and stems and leaves. And it's not just it isn't necessarily incorrect, is what I'm trying to say. It's just more correct if you understand that we really have a technical definition for what's a root, a stem, and a leaf. And these organisms can't have those things because they aren't the organisms that do have them. They aren't plants. All right, so you help me in the upper left. Answer this question for me. The fact that these laminaria does have a root-like thing. Good job, Isaac. The stem and a leaf-like thing are these homologous characteristics or analogous? So Isaac from finance, tell us why they're analogous. Isaac, tell us why they're analogous. Or if anybody else would like to give Isaac a help. No, they did not evolve from plants. One more time, and they didn't come from the same common ancestor. There you go. Okay, now remember this whole term of homologous, analogous. Okay, technically, right? Technically, we're all homologous. Technically, we all came from a single common ancestor, that universal common ancestor, right? Technically, we all came from that. But what we're talking about when we're using the words homologous and analogous is we're saying, do the structures look alike because of similarity of descent? They have a recent common ancestor, or do they look alike because they have a similar environmental pressure? So they were both shaped the same by Mother Nature, by natural selection, right? So this is a protozoan, and it may have a thing that looks like a root, but it's not a plant. As a matter of fact, it's not even one of the protozoans that are closely related to plants. So these structures are definitely analogous. They're not homologous structures, okay? So the similarities are simply because they both, this has got to have something to hold on to. It's got to have something to help it grow upright. It's a photosynthesizer. And the blades have to be held up or be approached to the sun, like in these big kelp forests. That's why it says here, you find them in relatively shallow water. They got to be able to get to the surface to be able to photosynthesize. Now, they got these cool little things, these things called nematocysts. And these nematocysts are actually floats. There's air is trapped in there and allows these things to float up. Very cool critters. They make these, especially out in California, make these very dense kelp beds. When I was stationed out there in the Navy, I would go dive. Water's colder than heck. But we would dive in these kelp beds. Really kind of cool because there's all kinds of little organisms, like a little juvenile nursery, lots of little critters and big critters, and I'm trying to eat the little critters. There's a beluga right here. Beluga, what a beluga does, and these are mats of seaweed. What beluga does is wait for some little fish to kind of swim out of the protection of the seaweed a little bit too far, and it snags one of them. Friends of mine who were divers in California when diving the kelp beds, some of the folks that actually live there and have been there for years said that mako sharks are basically miniature white sharks, miniature great white sharks. Great white sharks, the great white sharks are extinct, great white. The ones today, great white sharks were the ones you can buy those big teeth sometimes, like at rock shows. White sharks are what's alive today, but mako sharks, which are their relatives in California, what they'll do is they know there's a lot of those little meaty little fish hanging around out there in the kelp forest, but them swimming slowly through there, they would be knocking that kelp aside. The fish would see them coming a mile away. So what they do is they dart through these kelp forests very quickly, these mako sharks, and they're built for speed. They blaze their way through, trying to catch some fish out in the open and grab it and eat it. So the interesting thing is when you're diving in the kelp forest, and I would love to have seen something like this. I never did. But these buddies of mine would tell me that you'd be sitting there diving, floating one time, and boom, coming right out of nowhere would be this mako shark, slicing its way through the kelp, maybe 10, 15 feet away from you. Yeah, that's the great white shark. Carcharodon megalodon, or megalodon. Carcharodon kerkerias is the white shark that's alive today. Okay. Questions on brown algae and homologous, analogous, and holdfast and all them things that we're talking about. And we're going to talk about this next. This is a form of reproduction that you may or may not have seen before. You might have saw in Bio 1, or you might have saw it in some other biology classes, but this is a very unique form of reproduction. So let me show it to you. This is going to be laminaria. So this is the plant in this picture that we just looked at. This is how it reproduces. And it's not just laminaria that does it this way. Many protozoans and a lot of plants, many of the species of plants that we talk about, especially the mosses and those kinds of plants, reproduce this way. So this type of reproduction is known as the alternation of generation. And I want to walk you through it, and then I'm going to want to ask you some questions about some differences you might see between the way these organisms reproduce and the way we reproduce. Let's start with the diploid side. Let's start over here. The diploid generation, or the diploid structure in laminaria and in all organisms that have alternation of generation, is known as the sporophyte. Okay, and the sporophyte is diploid. You follow the cycle here. The pink is the diploid stages. The blue is in the haploid stages, right? So we're going to the same thing that we talked about in reproduction with humans, right? Meiosis produces haploid cells, and then fertilization produces diploid cells. Okay. So sporophytes have sporangia, and sporangia, they produce spores, and they produce those spores through meiosis. Now, what does meiosis do? What's the purpose of meiosis? Yes, diploid to haploid, but beyond that, or in addition to that, what is the purpose of meiosis? There were two major functions of meiosis. That's mitosis, right, Isaac? Mitosis. Yes, it's for reproduction, Harrison, you're right, but I'm actually going back to there you go, mixing genetic material. Yeah, we're reducing the diploid number to haploid, Jared, but what Melanie said, we're mixing the genetic material, right? Melanie, where's one of the places that happens in meiosis? Where do we set up that to mix up the genetic material? There's more than one place that occurs. By what event, I guess, is what I'm trying to get at. Right? It happens during prophase 1. Remember when homologous chromosomes seek each other out and they lay next to one another and they cross over, they form chiasma. There you go, right? Chiasma is shuffling up genetic information, right? And then the other part is where they line up in metaphase 1 or how they line up in metaphase 2, on the left or the right. All of that, meiosis is variability, folks. Mitosis is quantity. Meiosis is variability. That's why meiosis, exists, if you will. We're making cells variable. Now, in the case of sporophytes, like in laminaria, you can undergo meiosis a lot of times, and you produce a lot of these spores, these haploid spores. They're called zoospores because they can move, because they're flagellated. Some spores have a flagella, some don't. The word zoo normally means something to do with animals. These are obviously not animals. What we're tying into the idea that most animals exhibit the characteristic of motility. They can move. So zoospores have a flagella, so they can swim around, okay? All right, so you get a lot of these zoospores, these genetically variable zoospores, because you don't know what kind of environment you're going to have out there. And those zoospores disseminate into the environment, and they form these haploid structures, these haploid generations called gametophytes. Now, right away, there's a difference between this kind of reproduction and the reproduction that you learned about with human reproduction, right? We don't do that. We don't make spores. We don't make gametophytes. During meiosis, we make gametes. Now, you'll notice if you go down the circle a little bit further, there is gametes, egg and sperm. But before that, there's these things right here. In alternation of generations, when the zoospores germinate, what they do is they form a multicellular gametophyte, a multicellular haploid structure. That's where the term alternation of generation comes from. The sporophyte is the diploid generation. It's multicellular and it's diploid. The gametophyte is multicellular and haploid. And sporophytes make spores and gametophytes make gametes. And then gametes fertilize, and we're back to what you recognize, right? It's the zygote. And of course, then that zygote grows by mitosis into that sporophyte. So what's the big difference here in alternation of generation? What's the big difference here between that and the way, say, for example, human beings or typically other animals reproduce? Anybody? What's the difference here? Think about the way we reproduce. Think about what happens after we undergo meiosis. Do we have two multicellular generations, one diploid and one haploid? No, we don't, do we, Dustin? Right? Our haploid stage, is it multicellular or single cellular, unicellular, the haploid stage that we have? Kaylee? Anybody else? Jalen? Rayanne? Our haploid stage, okay? The sperm or the egg. Think about it for a second. Sperm or egg, are they unicellular or are they multicellular? You may make a lot of sperm, okay, or you may ovulate a lot of eggs, but each egg and each sperm are unicellular, right? They're unicellular. That's right, Jalen. Each one is unicellular, and then a unicellular egg and a unicellular sperm unite for fertilization. Here, we've got a multicellular gamete stage. We have a multicellular haploid stage. We have another entity, a thing, a generation that grows, and it makes lots of cells, and it's a multicellular haploid gamete producing factory is what it is, folks. Again, these are the critters that are going to deal with some of the harshest conditions, especially back in evolutionary time. So to compensate, what they're going to do is make numbers. So instead of undergoing meiosis, X amount of sperm or eggs or whatever, they get these multicellular gametophytes, and then through mitosis, they produce tons of egg and sperm. So you've already got the variability back in the meiosis part. So now the gametophytes are nothing but these, you know, the variability is instilled, already there, and you're going to make a lot of these eggs and sperm. Because what's going to happen to most of those eggs and sperm, folks? What's going to happen to most of the egg and sperm that are produced by these gametophytes? Yeah, they're not going to make it, are they? So you're going to overwhelm that with numbers. So the alternation of generations technique, if you will, or system is a way to produce a lot of gametes, okay? A lot of gametes, because you know a lot of those gametes are going to survive. It's also a way to produce this intermediate you know, stage that can stick around if things aren't really ready quite yet. Because some of the spores, not so much in laminaria, but in some of the spores of the critters we talk about, they can be, they can lay around for a while. So you've got this little sort of waiting stage. All right, so we're going to talk more about alternation of generation, especially when we get into plants in the subsequent chapters. If the generations don't look alike, like for example, laminaria, the sporophyte looks very much different than the gametophyte. We call it heteromorphic. And if the generations look very similar to one another, we call it isomorphic. Okay. All right, any question about alternation of generations? The big take-home message here with alternation of generations is it differs from us because it has a multicellular haploid stage. We don't. Ours are unicellular. Okay, the A in sarclade stands in the sarclade stands for the alveolates. There are three of those: the dinoflagellates that we're going to talk about, the something called the apocomplexans, and then the ciliates. The ciliates are the many of the critters that you guys are probably familiar with, right? The cilia critters, the paramecium, and all that sort of stuff. No matter which one of those groups we're looking at, all alveolates have share something in common. They share these sacs you can see in red in the picture I showed you there. On either side of the flagellum in this particular critter, these alveoli sacs. Now there are lots of things out there that have alveoli. You have alveoli. Where would we look in our bodies to find some alveoli or an alveolus? That's right. What are the alveoli in our lungs, Melanie? If you want to just kind of give a little description is what I mean, you know, just very quick. Yeah, air sacs. They're air sacs. They're where the air exchange occurs. So alveoli means sac. And if you look at here in the alveolates, they all have these flattened, they're calling them vesicles, but it's the same thing. It's these flattened sacs, these alveoli. Now, the interesting thing about the alveoli, what's even kind of interesting to me, is when I first started teaching this class, I used to say, we don't know what the alveoli do. Now we're beginning to gather more and more evidence. We think that the alveoli may function similar to Golgi bodies. And if you remember Golgi bodies in Bio 1, Golgi bodies exported material out of the cell. It might be easier if I wait to explain or to show you a critter that uses its alveoli, we think, to accomplish a purpose. And you'll see what I mean by exporting something outside the cell. So all alveolates have the alveoli. One of the alveoli that you may have seen here in Florida is one that causes this. This is red tide. Red tide is not caused by an algae. It's caused by a dinoflagellate. Now, a dinoflagellate is this cool little thing. It's got two flagella, one that goes this way across the body, and then one that goes the other way across the body, and it moves by spinning or whirling. That's how it moves around. That's where the dino comes from in dinoflagellate. It's a whirling flagella gizmo. Most are photosynthetic. They may not use the same pigment as plants. They may use the like in the red algae can photosynthesize, but they don't use the same pigment as plants. Unicellular with shells, just like in the diatoms, but not silicon dioxide shells, cellulose shells. Oh, cellulose. Who else has cellulose? What other organism we're very familiar with has cellulose? Yeah. So dinoflagellates and cellulose are closely related. So is that homologous or analogous? I might have said something that wasn't true, by the way. Cellulose and dinoflagellates, cellulose and plants. Homologous, analogous? Analogous. Absolutely. Dinoflagellates aren't closely related to plants. It's just that they both happened upon using cellulose. Cellulose is just, remember, a polymer of glucose, and dinoflagellates are exposed or have access to glucose, of course, so they can make cellulose as well. Okay, these are aquatic organisms who are planktonic. That word planktonic has a very specific connotation. It's oftentimes used to refer to many different kinds of critters, but if you're a planktonic, you're a drifter. I think it literally means that from Latin. Planktonic is drifter, nectonic is swimmer. So fish, marine reptiles, sea turtles, marine mammals, things that can move through the water are nectonic. Things that float are drifters. And dinoflagellates are drifters. They may be able to move a little bit with their flagella in kind of a localized area, but they're pretty much at the whim of the currents, right? Probably one of my favorite things about the ocean, or my favorite things of memories of being at sea in the Navy, was the luminescence of the sometimes bacteria, but also dinoflagellates. Many dinoflagellates have these bioluminescent bacteria that live in symbiosis in the tissues, or not in the tissues, but in with them. And when you agitate these bioluminescent bacteria, they go through a chemical reaction and actually give off light, sometimes a green or sometimes a bluish color. And it'd be so neat at night if the ocean was flat, the ship was cutting through the water in the wake, and if we ran through a bunch of these dinoflagellates or these bacteria, it would just glow. The wave coming, that V coming up the bow of the ship, it would just glow blue or green. It was absolutely incredible. You can find all kinds of neat videos online that shows you that sort of stuff. Okay, so that's an alveolate. Let me show you the alveolate that I was talking about. That what happens in red tide is a toxin. The dinoflagellates that cause the red tide bloom, so they go crazy and start reproducing, because of the nutrients that are there, or environmental conditions, and they release a toxin, and that gets into fish. Marine mammals who eat the fish can die. It bothers mucous membranes, so sometimes that's why if you're at the beach or in a red tide, you'll notice there's a lot of coughing going on, because the spray is spraying that aerosol and that toxin, and it's irritating your mucous membranes. Okay, so the alveolate, who we give some idea of what those alveolar sacs are about, are the apical complexans. And the apical complexans are animal parasites. And down at one end, near the apex, near the point edge, there's a picture of a cell, of an infectious cell, a merozoid. We'll talk about what a merozoid in just a second. But down near that apex, there's an apical complex, okay? And that's an arrangement of organelles in conjunction with those alveoli down there that we think has something to do with allowing it to invade the host cell. So the fact that those alveoli are like Golgi bodies, they might be secreting some material or producing some material that allows this merozoite cell to drill with its apex into the host cell. And the host cell in this case, you can see, is the red blood cell. This is what the malaria parasite does. The malaria parasite, or the Plasmodium parasite, uses a series of infectious cells. I don't really want to call them larvae, because they're not. They're infectious cells, merozoites and sporozoites, and we'll look at that in just a second. Goes through this very complex life cycle. A lot of parasites have these really complex life cycles. It's always interesting to me. I worked on one when I was in the lab in South Carolina that infected fish. In one stage, it infected ducks and snails, and a specific species of snail. So with these parasites that are very specialized, they're kind of, they got to be careful, because if the snails die off, then they wouldn't be able to reproduce. So it's kind of like the koala bear only eating eucalyptus. They're very good at what they're doing, but they are sort of walking a tightrope of specialization there. So this Plasmodium parasite has very complex sexual, asexual, different kinds of hosts that are necessary for it to divide and survive. And I'm going to show you that in just a second. But malaria was a scourge for many years, especially in the tropics. And getting rid of the mosquito was the trick, right? It's the vector. I'll show you why the mosquito is the vector, or how the mosquito plays into this. Now, though, we've been spreading the mosquitoes with DDT was very helpful, but unfortunately DDT was also ruining the eggs of songbirds. DDT gets into the food system, and birds that eat fish or eat fish that eat fish, or however it occurs, accumulate DDT in their bodies, and DDT messes up what's called the shell gland. And in birds, the shell gland is what lays the shell, obviously, around the eggs. So birds with DDT concentrations that were too high were laying eggs with abnormally thin shells. So during the incubation period, when the mother's crawling around and trying to keep the egg warm, she would often break it, and of course the embryo would die. Get rid of the DDT and the bird population survived. But unfortunately, DDT was used overseas, and I think in some cases still is being used overseas. And then we run into the problem of DDT resistance too, for reasons that we've talked about. So drug development and vaccines are probably, one of the best ways. Let's treat it at the end of the human side of it rather than try to destroy the ecosystem. All right, let's look at this complex reproductive cycle of the mosquito. And we'll start in the upper left with the mosquito on your skin. Now, mosquitoes are blood meal parasites. So like all blood meal parasites, like ticks and fleas, very often when they puncture into the capillaries, they're going to secrete a little bit of a salivary juice, if you will, but an anticoagulant, okay? And an anticoagulant is going to keep the blood flowing so that the blood easily passes into the parasite, and the parasite moves on. Now, when a special type of mosquito, the species is actually the Anopheles mosquito, and I always spell this wrong, so I'm going to cheat and look in your book. The Anopheles mosquito, when it dumps a little bit of that salivary juice, that anticoagulant into you, in its salivary glands there are these sporozoites, and sporozoites are infectious cells of the parasite, of the Plasmodium parasite, of the protozoan, not of the mosquito. Sporozoites actually form from something called an oocyst, which is found in the lining of the gut of the mosquito. So an oocyst that's formed from the zygote there through meiosis forms these variable sporozoites, okay? Now those sporozoites are then dumped into the bloodstream of the human while the mosquito's feeding, and they make their way to the liver. In the liver, those sporozoites become and transform into another type of infectious cell, the one you saw in the previous picture, the merozoites. And merozoites infect red blood cells. What an incredible, look at this neat life cycle here. You got the mosquito, you got the human, and you got two parts of the human that you need. You got to have the liver to make merozoites, and then the merozoites go out and infect red blood cells. And what they do is they asexually reproduce in the red blood cells. When enough of them form, the red blood cells lyse or rupture. That's what causes the fevers and the chills. Remember Wallace? Remember he was talking about during a malaria fever and a chill episode, he came up with this idea of natural selection, okay? So that's what's causing that. Now, the reason why the merozoites are infecting red blood cells is some of those merozoites, when the red blood cells lyse, have changed or transformed into another type of cell called the gametocytes. And gametocytes complete the cycle, all right? Gametocytes are then transferred back into the mosquito when it takes in a blood meal, and then the male and female gametes, then, of course, fertilization occurs. Now, the male gamete isn't that star thing. It's the little worm things all around it. So the male gametes makes tons of these male gametes. And then it fertilizes, and we're back into the zygote. So we get the variation in the meiosis, okay, so that we have different kinds of sporozoites. And then some of those will survive and make their way. And then, of course, all of this reproduction in the red blood cells of the merozoites, this is all asexual. But all you're doing there is cranking out numbers. Okay? Parasites are weird. You look at them sometimes and they have parts of their life cycle that just seems like, what exactly is that supposed to do? But it's all evolved, co-evolved together throughout years. Very neat. Any questions on the life cycle of the mosquito there, or the Plasmodium parasite? The mosquito again being the vector. And the last group of the alveolates are the ciliates, and they're called that because they have lots of cilia. And cilia are nothing more than just basically shorter flagella. And organisms usually have lots of cilia, right? Remember from Bio 1? Generally, you'll have a lot of cilia, few handful of flagella, and generally flagella are larger. So paramecium, this is a paramecium here. This looks like the bottom of a shoe. It's like the sole of a shoe. So it's surrounded by cilia, has a contractile vacuole. Contractile vacuole, which gets rid of excess water. So we can do a little detective work here again. If paramecium have a contractile vacuole, remember Euglena had it too, what type of an environment, tonicity-wise, what type of environment do they live in? Do they live in an environment where they have to worry about losing water or gaining water? And what happens to cells in a hypotonic environment, Melanie? Think about it again, Dustin. It's a contractile vacuole. It's getting rid of excess water. Contractile vacuoles are going to be like the bilge pump in a leaking boat. They're going to get rid of the excess water. So in a hypotonic environment, water flows into the cell. In a hypertonic that it flows out, folks. So again, that's how when we look at organisms, especially like extinct species in the fossil record, if we can identify a contractile vacuole, let's say, we can surmise that that organism, it's using that inductive and deductive reasoning thing again, that probably lived in a hypotonic or freshwater environment. It wasn't a marine species. Now, they're not photosynthetic, so they do have a mouth. Really kind of cool on a microscope. Sometimes you can see them spinning and turning. You can see their mouths. They're really kind of neat critters to watch. But I want to talk about variability again. So I want to talk about what this whole micro and macronucleus thing is all about. They got two nuclei. So let's see what they're going to do with those two nuclei. Ciliates or paramecium can shuffle up genetic information by undergoing conjugation. And again, conjugation is like we saw in the prokaryotes. Two of them get together and they form that sex pili. So they're exchanging genetic information. It's kind of like sexual reproduction. The only reason we don't really call it officially sexual reproduction, if you look up here, number 1, we don't say parameciums are different sexes. We say different mating types. Paramecium don't really have sex. They don't have little genitals to look at, right? Obviously. So what we're saying is we got paramecium of different mating types come together, and we're going to indicate those by different colored micro and macronuclei. So they come together, and meiosis then occurs. Here they are in conjugation, by the way. So meiosis occurs in the micronuclei, not in the macro, but the micronuclei undergoes meiosis. And you remember meiosis, you have one cell that divides and divides again, so you're going to get 4. So that's why there's 4 in that picture there. At the end of those meiosis, and you get those, now there's the variability part again, and you get those 4 haploid micronuclei. At random, 3 of those are going to degenerate. So at random, 1 is left, if you want to look at it that way. And now that's going to divide by mitosis. So we got the variability in meiosis. We got the variability in which ones degenerate. Now we're going to divide them in mitosis. You're like, why meiosis? Then get rid of 3 and then divide them again? Well, this is the swapping part. The reason why you make a copy of the micronuclei is the micronuclei, they're haploid, you see. And in conjugation, you're going to swap one of your haploid micronuclei for one of the ones in the other cell. See, the red goes to the purple and the purple goes to the red. And then the micronuclei you have is going to fuse with the micronuclei you got from the other cell. The macronucleus is going to disintegrate. The micronuclei is going to divide by mitosis. One of those is going to become the macronucleus. One's going to become the micronucleus. So it is kind of sexual reproduction in the sense that you're mixing up 2 genetic organisms' information, but it's not sexual reproduction in the way that we typically think about it with eggs and sperm, right? But it still basically does the same thing. It shuffles up genetic information. And of course, paramecium can also divide asexually, just by dividing in half, right? But this, that was going to give you clones. This is actually, and it's kind of neat the way that works, shuffling up that information and passing those nuclei back and forth. Oh, good. Perfect time. All right. Any questions on that, folks? All right, let's go to this last group in the SAR clade. And the R stands for rhizarians. Okay. Many rhizarians are amoebas, but there we go. That word amoeba is not monophyletic. So things that are called amoebas may not necessarily be a taxonomic amoeba, right? Understand what we mean by that? We've used that term enough. So the word, and I put in quotes, many rhizarians are amoebas. Some amoebas are monophyletic, but just because something's called an amoeba doesn't mean it's related to other amoebas. Doesn't mean it has a close common ancestor. But all rhizarians have pseudopods, okay? They have these thread-like pseudopods. Other amoebas that you might be familiar with have lobe-like pseudopods, and they're different kinds of amoebas, okay? So that's the amoeba that's not monophyletic. So rhizarian amoebas are monophyletic, but if you're talking about the amoebas that with the lobe, the ones that you may saw in Bio 1, that's in another group of protozoans. How mixed up this whole thing is. But all rhizarian members of this group have these thin thread-like pseudopods that stick out. You can see that in the picture down there. Radiolarians fit into this group. See, here's a radiolarian. Go. And look, radiolarians have a silica skeleton and then these pseudopods that stick out. Now, these are marine organisms. They live in the ocean. This is another picture. This is the marine or the silica skeleton or the test. The pseudopods stick out through little holes in the tests. Now, the pseudopods do two things. They're used for feeding, like they use to capture organisms and then, to digest them. And they're also used, if you've ever been on the ocean and try to float on your back, you know that the key to floating on your back is to try to present a large surface area, right? You're trying to get as much resistance to the water as you possibly can. It's called drag. Well, radiolarians, marine organisms living in the ocean, they don't want to sink. Many times, some of them are photosynthetic or have photosynthetic partners that they work with, so they want to stay near the surface. So what they do with these pseudopods, those pseudopods increase drag. Those pseudopods, a bunch of those little needle-like things sticking out, increase the resistance, so it's much more difficult for radiolarians to sink down through, or rhizarians in general, not just radiolarians, to sink down through the water. Clever little critters, aren't they? Okay, now the next group of critters has the formidable name of the foraminiferans, or if you're really cool, you call them the forams. Anybody ever seen a foraminiferan? Anybody ever seen a foraminiferan or a foram? Kaylee, two Kayleys, Rayanne, Melanie, Bradley. Not sure. Okay. Isaac, you ever seen a foram? Bradley, how about you? Tiffany. Harrison, you've seen a foram, haven't you? You've seen a foraminiferan? I bet, well, now that I think about it, maybe you all haven't. I was just getting ready to say, I bet all of you have. But it depends on how long you've been in school. Yeah, kind of a trick question. I remember when I first started teaching this class, there were foraminiferans actually in every classroom. Whether I was teaching science or math or whatever, there was always foraminiferans. But what do I mean by foraminiferans and the fact you've seen them? Chalk. The white chalk in a classroom are the tests or the shells of the next group of rhizarians, the foraminiferans. There's a picture of a foraminiferan, thread-like pseudopods coming through a shell, but now the shells are made of calcium carbonate. All right, so we've had shells made of silicon dioxide, cellulose, silica. Now we've got calcium carbonate. That's the white of the chalk. That's when on an old chalkboard, that's what that is. Those are the shells of foraminiferans. Foraminiferans are like the laminaria. They have the haploid and the diploid life cycle. The picture you're looking at are the White Cliffs of Dover, which are on the east coast of England. So what this is a bunch of foraminiferans. Foraminiferans are heterotrophic marine organisms, so they live in the ocean. And throughout many, many millions of years, they died and fell down to the bottom. And then through geological or tectonic events, the bottom of the ocean was lifted back up. So the cliffs, the White Cliffs of Dover, were at one time the bottom of the ocean. And all the white in there are all the shells of those, oh my God, what, billions and billions of foraminiferans that died throughout the years and drifted down and settled on the bottom. So it maybe was a little bit of a trick question because I don't think anybody's used chalk and a chalkboard. Like I said, when I first started teaching this class, I actually did use chalk. The old green chalkboards. Or were they black? I don't remember. Okay, the last group that I want to talk in the rhizarians, I think this will be the last group I talk about today, be a good place to finish off, are the critters we call the cercozoans. Small little group, very diverse morphologically, right? They're very difficult to, as far as like group them together just on the way they look in appearance. So that, again, that's why genetic analysis is really helpful in this day and age. They're found everywhere, though, in water, the marine, freshwater. Move around through flagella or pseudopods, and they're an interesting support for the eye of endosymbiosis again. Remember I showed you the nucleomorph earlier? Well, this is an example of a cercozoan. This is Polynella chromatophora. Okay, Polynella chromatophora. And this is what Polynella chromatophora looks like. This is its flagella. And Polynella chromatophora, as you can probably imagine by looking at it by its green color, is photosynthetic. And it's photosynthetic because of this organelle, or this part inside of it that's known as a chromatophore. Okay, that's where the name Polynella chromatophora comes from. This is the chromatophore, this green thing. That's just part of the organism. When they looked at the chromatophore a little bit more closely, they found that the chromatophore was surrounded by a peptidoglycan membrane. Now think about that for a second. Think about what I'm saying. Where did we see peptidoglycan membranes? Who is the only group of living organisms that have peptidoglycan in their membranes? Yeah, right, the bacteria. Archaea don't have peptidoglycan, but the gram-positive, the gram-negative, either it's thick or thin, it doesn't matter, you have peptidoglycan. So if that endosymbiotic theory that we've looked at is some eukaryotic cell engulfed a bacterial cell, then here's some really strong evidence. We see it in Polynella chromatophora is alive today. This is a living organism that shows evidence of recent, and relatively speaking, recent endosymbiosis. I mean, the endosymbiosis has been so recent that this chromatophore still has that peptidoglycan membrane around it as if it were still a free-living bacterial organism. So it's a lot of detective work in science that occurs sometimes, and that's why, was it Newton who always said chance favors a prepared mind? If you know what to look for, then sometimes you get these neat little clues, and you go, wow. You know, you can imagine the scientists first looking at Polynella and examining and going, why does that chromatophore have peptidoglycan? That's a substance that we've only ever seen in bacteria. The best explanation about that, the most parsimonious, is that it was a bacteria at one time and it got engulfed through endosymbiosis. Okay? All right, well, I believe that's my time for today, folks. So next week when we get together, we'll move on and talk about, I think, the archaeoblastids on the next one. Yep, so this is that weird group that's got protozoans in it mixed in with things that are definitely not protozoans. So, okay, so I'm going to stop the recording.